We have been talking about transmembrane proteins. We have looked at example of membrane protein that transverses the membrane single time. We know there are many proteins that transverse the plasma membrane more than one time. So, for example, we have talked about G protein coupled receptors. These receptors have 7 transmembrane domains. So, how do cells insert these membrane proteins which transverse membrane several times? So, let's look at that. In multi-pass transmembrane proteins, the polypeptide chain passes back and forth repeatedly across the lipid bilayer. An internal signal sequence serves as a start transfer signal in these proteins to initiate translocation. So the proteins we have talked about so far, their start transfer sequence is at the end terminal of that polypeptide chain. So when that sequence emerges, the protein synthesis is halted and the ribosomes, unless and until till they dock at ER, at a specific location at ER, protein synthesis does not resume. So in this case, what I'm telling you is that start transfer signal is actually not present at the end terminal, but it is present somewhere in the middle of the protein perhaps. Once the signal comes out of the ribosomes, the same thing happens. Signal recognition particle will attach to the complex and halt protein synthesis till the ribosome docks on the surface of the endoplasmic reticulum. Let's look at a simpler example to begin with. We'll talk about the double pass transmembrane proteins. I'll show you how it works. Here, for example, you can see this is a protein which has 2 sequences, this one in red and this one is orange. So as you know that first of all, the amino terminus comes out of the ribosome. So this is the first signal which is going to serve as a start transfer sequence, and once this comes out, the production of protein will halt till this protein is actually inserted in the Sec61, the translocator. Once that happens, the protein synthesis will resume till the second sequence comes out. This is again the sequences, both these sequences are similar. Basically, they're hydrophobic amino acids. So once this comes out, this sequence will also be sequestered by Sec61, the translocator, and rest of the protein will continue its synthesis. So once the protein synthesis has completed, the Sec61, the translocator will open laterally and the protein will be released in the membrane. So this protein happened to have 2 transmembrane sequences which are now embedded in the membrane of the endoplasmic reticulum. I would like to demonstrate you. We are going to use this cord as our protein. So protein synthesis will halt once this blue domain comes out. This is the start transfer sequence. So once this is exposed, the particle, the signal recognition particle, will grab this whole complex, this little piece of protein along with the ribosome, take it to the endoplasmic reticulum. Once it docks, the protein will be inserted through the Sec61 translocator and rest of the protein synthesis will resume. So once that happens, the protein synthesis resumes. The next sequence, which is also a hydrophobic stretch of amino acids, both these sequences are very similar. So once this comes out of the ribosome, again the signal recognition particle will bind it and also grab it and take it to the Sec61 translocator. So rest of the protein synthesis will continue, and once the protein has finished synthesizing, the Sec61 will open laterally and this membrane will be released in the membrane, endoplasmic reticulum. So now you can see that there are 2 domains, transmembrane domains, that are present in the endoplasmic reticulal membrane. So let's continue and see how our multi-pass domains, which with more transmembrane segments, is inserted. In more complex multi-pass proteins, in which many hydrophobic alpha helices span the bilayer, a second start transfer sequence reinitiates translocation further down the polypeptide chain until the next stop transfer sequence causes the polypeptide release, and so on for subsequent start transfer and stop transfer sequences. So here, for example, you can see this protein has 1, 2, 3, 4, 5, 6, 7 transmembrane domains. All these transmembrane domains is basically a stretch of hydrophobic alpha helices. Now, the orientation basically in this case depends whether a sequence is going to serve as a start transfer sequence or stop transfer sequence. Basically depends upon the location of this particular stretch. So here, for example, the first stretch of hydrophobic amino acids is going to serve as a start transfer sequence. Subsequent stretch of hydrophobic alpha helices is going to serve as stop transfer sequence. And then the next hydrophobic sequence will be start transfer and stop transfer. It goes on sequentially till the protein with multiple transmembrane domains is inserted into the plasma membrane, or initially, of course, in the membrane of the endoplasmic reticulum. So we have seen how proteins with multiple transmembrane domains are inserted in the endoplasmic reticulum initially, which from where they will be enclosed in a vesicle which can be transferred to the plasma membrane, for example.